Okay, so let's start. So first of all, hi everyone. Thank you all for joining me, and as well as the online participants. And thank you, Pietro, for the kind introduction. So today's talk is the second talk in a series of 3 talks that I will be giving over here. And today's talk is titled Incorporating Prior Knowledge into Continuous Flow Matching for Dynamical Systems. So last time we saw what flow matching is, what the essence of flow matching is, how the algorithm works in the sort of the inner workings of the algorithm. In this lecture, what we are going to look at is what are the critical design choices that flow matching makes and what are the critical knobs that you can tune, especially in the context of dynamical systems, to make flow matching work better. So just to recap and just to summarize, what is flow matching? What is it doing? So like we saw last time, flow matching is basically characterizing the rate of change of a random variable that starts from an initial tractable distribution, can be Gaussian, can be anything, and eventually by characterizing how you take this random variable, apply the set of continuous transformations such that in the end it ends up at a complicated distribution that represents your real world data. So characterizing this whole sort of transformation in a simulation free manner is what flow matching is all about. And in the end, once you characterize this complex distribution, you can sample based on your training data, you can sample things like small molecules, proteins, images, music, or even like a sequence of embryogenic states, depending on what you train your model on. So, and that's what essentially all generative modeling is all about. And flow matching is basically a recent addition in this line of thought. It's simulation free and it's efficient and it's elegant. And compared to diffusion-based models, it's a ODE formulation, not a SDE formulation as happens in diffusion. So it all boils down to, as we saw last time, it all boils down to defining your conditional probability paths, because the main sort of essence of flow matching is that the set of transformations that you want to characterize are hard to characterize. But let's say if somebody gives you an easy way for characterizing this transformation, let's say you start with initial state and you just draw a straight line, and that straight line ends up at a data from a real world training distribution. So this is like a simple transformation as well. And so can you use these simple transformations and use all of these simple transformations to then end up with a neural network that basically encodes these sort of conditional paths that you've constructed yourself, such that the marginal from these things, which is encoded in the neural network, then gives you the true distribution during sort of inference time at time t equals 1 or during sampling. And the characterization, the simple characterization of these tractable conditional probability paths is extremely critical. And as we saw last time, in the case of you assuming the fact that this transformation is a Gaussian transformation, and assuming that you are also using sort of linear interpolants as your conditional probability paths, the nature of the conditional velocity field and the conditional probability paths looks as the following. And here also you're making like a choice in terms of how you couple your initial and final state. And we'll come back to what coupling choices make sense, essentially. And that's in essence what this slide is representing you, that you had some initial state and final state, and now you've constructed straight conditional probability paths by basically drawing a straight line over here. And this is usually like a choice that people make, but although this is not the only choice, and in most cases it's not the right choice as well, as we'll see further ahead. So yeah, so given that you've now constructed conditional probability paths, like what do you do with them? You train, long story short, it all boils down to training like a regression objective, where your neural network, which is representing the true velocity field, is regressed over conditional velocity field that you've constructed based on your sort of design choices. And commonly used ways to sort of construct these conditional probability paths are basically constructing linear interpolant paths. In the case of linear interpolation, the conditional velocity has a very sort of simple and tractable nature to it, which is just x1 minus x0 by 1. But it also depends. So just because it's a straight line doesn't mean it's easy. You also need to decide how are you going to couple these x1 and x0, because what you have essentially are samples from two distributions. So how you're going to connect them is also like a design choice that you make. And often people use optimal transport to couple these things, because it has a nice interpretation in terms of you drawing the conditional paths such that the transformation, the work done to transform a given set of samples into another set of samples is minimized, and it has been shown to work well in practice, although there are some downsides to it as well, as we'll see in the future slides. But the sum total of everything that I've said so far is the following. The main bottleneck in flow matching is the fact that you're training your velocity field on the conditional on the velocity fields that lead you to conditional probability paths. And because this conditional velocity field is essentially your regression objective, so it's natural that your quality of learned velocity field is kind of upper bounded by the quality of your conditional velocity fields. So if your conditional velocity field is wrong, then obviously your underlying flow is going to be wrong. And this becomes important for dynamical systems compared to like generative settings, because in generative settings, what you have is you just care about how you take a state that can be any state, that doesn't have to be noise necessarily, and how do you end up to like the state that you want. So you just care about the endpoint fidelity. So how good are your endpoint generation? But that's not the case with dynamical systems. In dynamical systems, what you're supposed to do is you're supposed to also truly characterize the path between observations as well. And so here, if your conditional probability paths and the corresponding conditional velocity fields are not representative of the underlying sort of dynamics, and you're going to train, and if you're training your neural network on these sort of implausible or false conditional velocity fields, then naturally what you're going to end up with during sampling time is implausible generation. And this is exactly sort of the essence of the talk that I want to convey. And this is exactly why we also spend efforts towards like designing better, more plausible conditional paths. So I'll start with the first part of this talk, which is spline flow, which is work I did with Pietro and Xiao, my supervisors. So going from generative to dynamical systems, like what changes? So in generative settings, you're primarily concerned about going from, let's say, noise to data, and the corresponding conditional probability path has the nature as written here, and similarly the corresponding sort of velocity field can be simply characterized as well. But in dynamical settings, you just do not have 0 to 1. You basically have samples that are spread across a time horizon. And so how do you characterize conditional probability paths when your observations are spread out across a time horizon? The simple way to do so is by basically just doing things step by step. By step by step, what I mean is that if in generative settings you're going from noise to data or data to data, then in dynamical settings, if you have time going from T1, T2, T3, T4, and so on and so forth, then you just consider the fact that you're going to generate things between T1 and T2, and then T2 and T3, and then T3 and T4, and so on and so forth. And so once you stitch these linear paths, that's usually what people sort of also go for as their first try. And that's essentially what this equation means, that you're basically stitching these paths together and training your velocity fields on this stitch. And dynamical settings are everywhere. I would say that dynamical settings are the most general formulation as far as generative modeling is concerned, because think about video generation or world models that people recently talk about, or single cell evolution dynamics, or the conformations of proteins and how protein is interacting once it comes in the vicinity of a substrate or a ligand or things of that kind, or weather forecasting. So the potential is obviously huge. And this is what it essentially looks like. So let's say you have, in the example over here on the left, you have a video frame in action, which is basically just a video which takes a blob of sand, and this blob of sand is then translated into a S shape sort of final frame. So how do you move these individual particles such that you end up with this shape? And so characterizing this whole process dynamically is basically what dynamical system is all about, so to speak. And similarly for protein conformation dynamics, given that the protein was in some initial state, and now let's say you brought this protein in some electric field in the vicinity of a ligand or a substrate, how is that protein going to unwind and going to move in practice? You usually run like DFT simulations for these. And so even this involves you characterizing the initial state of the protein and how it's basically evolving in time, so to speak. And so this is also an example of a dynamical system. Now, given that we've established the fact that dynamical systems are important and more or less like a generalization of generative modeling, why are previous design choices suboptimal in these cases? It's easy to see that the previous design choices, in the way that they are designing these conditional probability paths, by and large, they use straight line interpolation. And so you're basically stitching these temporal observations by drawing straight lines between them. And these can be suboptimal for the following reason. So when you draw straight line paths between your observations, what tends to happen is that the straight line in the intermediary between the observations is kind of representing an approximate dynamics, so to speak. Okay, and because this conditional dynamics that you've proposed is approximate, there's obviously going to be some approximation error with regards to the true underlying dynamics. Okay, and this approximation error is in essence what we want to bridge, so to speak. So you can see this in the form of an example over here. So what we have here is a damped harmonic oscillator system. So a damped harmonic oscillator is basically when you have a blob of mass attached to a spring and you release that blob of mass from a distance, and that potential energy is then basically converted into kinetic energy. And the damping here essentially comes from the fact that you also have friction with the contact surface. So over a period of time, you basically get a damping effect. But without going into the equations of the damped harmonic oscillator, what you have here on the right is basically the position that is varying with time. And the red dots here imply the observations that you're making. Okay. Now the point is to reconstruct the whole trajectory based on just these red dots. If you take a linear conditional probability path, or a linear conditional path in this case, you're basically going to draw straight lines between these observations. And hence you're going to miss out on things like these small curvatures. Okay. And that essentially results in big effects overall, because you're now training your velocity fields and on these conditional paths that have approximation errors, and these approximation errors, when taken as a whole in the training data, are going to sort of blow up your final velocity field, so to speak, which does not happen in the case of B-splines, and that is why we use B-splines. Now in the case that I've shown here, if you notice how the curvature is captured by B-spline compared to linear interpolants, it's very apparent that B-splines seem to be, although we haven't talked about them, seems to be very adept at capturing the true nature of the underlying dynamics and the true shape of the underlying function based on like sparse observations. And that's exactly why we propose sort of utilization of B-splines. So B-splines in essence are a more sophisticated way of doing this interpolation. So interpolation, and when you use linear interpolants in particular, that's not the only way in which you can basically connect a set of observations. Like that's a design choice, yes, but that's not the only choice. If you recall like your high school mathematics, this problem essentially boils down to the fact that if you have n roots of a polynomial, what is the simplest polynomial that you can, or the simplest polynomial that you can possibly construct? Okay. In the case of linear interpolants, it's not differentiable, but you can just as well take something like, if you have the roots as x1, x2, dot, 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 up till xn, then you can construct a naive polynomial as x minus x1, x minus x2, x minus x3, dot, 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 up till x minus xn. And this indeed is what a Lagrangian interpolation is all about. But these naive polynomial interpolations suffer from something called as Runge's phenomena, meaning that they are very unstable between the roots, so to speak. B-splines, on the other hand, are continuous. They are piecewise local polynomials of some order that you construct recursively using Cox-de-Boer recursion formula. And they have very good properties compared to like other commonly used interpolants, in the sense that they are smooth, they are stable, they are mathematically flexible, they are flexible in terms of the order of underlying dynamics that you want to characterize, and more importantly, they do not encounter this Runge's phenomena and can adapt based on the system's complexity. And yeah, and that in a sense is the conscious design choice, that the first conscious design choice that we are going to make in terms of using flow matching to model dynamical systems. Now the question that you might ask is, isn't flow matching like a generative modeling algorithm? Like, why are you using it to design or characterize or use it for dynamical systems in general? And that's a good question. And the answer to that is the fact that in essence, flow matching is basically a highly scalable and simulation-free way of training continuous normalizing flows, which in essence is all about learning the velocity field of a ODE dynamics. Okay, so all ODE dynamics can be written in this following equation over here, where the variable is changing based on some drift, corresponding to some drift, which is also called as the velocity field. And your whole point is in ODE dynamical systems to characterize this drift. What is the nature of this drift? And these variables can be sort of any dimension. What is the rate of change of these variables that gives rise to observation that you make, so to speak? These kind of problems are what neural ODEs are solving, which in essence involves learning the continuous normalizing flows, although using expensive adjoint methods. And because flow matching are in a sense a scalable and a simulation-free way to train these continuous normalizing flows, hence you can also use flow matching for training dynamical systems. Now, in terms of like mathematical guarantees, what do we have? Like compared to linear interpolants that people use and compared to the B-spline interpolants that we are proposing, why should we believe in the fact that these are the optimal choices compared to the previous ones? So the first guarantee that we have is in terms of approximation error, which is kind of a no-brainer at this point. So this theorem 3.1 from the paper is basically talking about the fact that if you have a set of observations and you're going to sort of draw interpolants between those observations or represent the true underlying function using an interpolant function based on these observations, then what is the approximation error between the interpolant function that you've devised versus the true function, so to speak? And as we saw in the case of the damped harmonic oscillator, the error in sort of linear interpolants is higher than B-spline. And mathematically speaking, you can show that in essence looks of the following form, where the approximation error of the B-spline is order n raised to minus m, where n is the number of observations and m is the underlying polynomial degree that you've chosen. And that for the linear interpolants, it's order n raised to minus 2, where n is also the number of samples observed, so to speak. So you can naturally see that as n becomes larger and as m also kind of becomes larger, you are going to get lower approximation on B-splines compared to like linear interpolants. And similar guarantees also translate to the derivative of these interpolants, so to speak. And these guarantees on derivatives are important because in the end, what you're going to train your velocity field is on the derivatives of these interpolant functions, so to speak. And so the implications are simply the fact that theoretically better approximation of underlying dynamics should kind of result in better characterization of the learned velocity field. And that in essence what theorem 4.1 is trying to do. It's trying to then say what is the basically mean approximation error for your learned velocity field and the true underlying dynamics, which is f dash of t, so to speak. And this is kind of upper bounded by the training loss, the square root of the training loss, and approximation term, which is order n raised to minus m plus 1. So again, if you train your model well enough based on right inductive biases, depending on the kind of data and a good training algorithm, then it's observed that in practice, the training loss usually goes to 0. And compared to sort of linear interpolants, then the error basically boils down to how well have you approximated the true underlying dynamics. And in the case of B-spline, because you've characterized the true underlying dynamics well, you naturally get a learned velocity field that's much closer to the ground truth dynamics, so to speak. And so the implications for this theorem are also the fact that better approximated conditional paths eventually lead to learned velocity field that's true to the underlying dynamics, and that in a sense is what we want. But all of this is not well enough. Like, these are good to have properties and these are the properties that we want, but we also want to see the correctness of the whole sort of pipeline in the following sense. Because you're ultimately using flow matching as a framework, you need to make sure that the guarantees of flow matching directly translate also to the dynamical case, and especially in the case when you're using B-splines. And what are these guarantees that flow matching gives us? These guarantees are in the sense that the conditional paths that you're going to characterize and the corresponding conditional velocity fields need to observe continuity equation. That is first. And the second guarantee is that you can recover your true marginal velocity field as well as the probability from the conditionals that you're devising. And this is also something that we prove for the spline case. And as well as the fact that once you do all of this, once you utilize these recursive ways of calculating interpolant functions, you also need to basically come up with a good characterization of the first derivatives, which is what you're going to train your model on. If not, then you can't use this framework. And so that is also what we do, in the sense that this is how the conditional velocity field looks like. And so all of these are tractable, and this basically reinforces the confidence that what we are doing is logically sound, at least. And that was just for ODEs, but you can easily extend this framework to modeling SDEs as well. Now, compared to ODEs, for the SDE case, what you have is a drift term as well as a diffusion term, this G of t over here, and dWt is basically the Brownian motion term. Okay, so the state evolves based on the drift as well as the stochastic Brownian path over here, and we can use the same sort of line of thought or argument for modeling SDEs for cases where we know what the diffusion schedule is, or we know that the diffusion schedule is kind of constant, so to speak. Only for these cases, we can definitely use the machinery that we've just built for the ODE case as well. And why are we able to do this? We do this by converting the SDEs to probability flow ODEs. Now, what do I mean by that? Now, when you have a ODE or when you have a SDE, remember that you are writing down these set of dynamical equations on random variables, okay? Even though those random variables may have like very, very low variance such that they kind of behave like not a random variable or a constant observation, but regardless, it's a random variable in principle. Now, given that it's a random variable, we naturally care about the probabilities of these random variables at each point in time. What probability flow ODE is telling you is that for a SDE that starts at some point and ends up at some terminal distribution, I can give you a corresponding ODE that will essentially have the same marginal distribution at time t, so to speak. So even though you are writing not a SDE, you're not writing a SDE, you're writing a probability flow ODE, the properties of this probability flow ODE are such that at time t, the marginal distributions that you would have gotten based on this ODE or the SDE would have been equivalent. And this is the property that we want. When we say that you are training your neural network velocity field or score function or whatever, what you essentially care about is when I use these drift or score functions in practice, do I end up generating samples that truly reflect the underlying reality or the training data? Yes, is the answer in this case, and this is why you can sort of convert the SDE to a corresponding probability flow ODE using the description over here, but I will say now. So the reason or how you can do this is by basically looking into the Fokker-Planck equation. So the Fokker-Planck equation is the continuity equation equivalent for the SDE case. So the Fokker-Planck equation is telling you, given that you have some SDE, what is the interaction between the probability as well as the drift term with time, so to speak, because it's a random variable that's evolving over time. So naturally the corresponding probability density is also going to evolve over time. So how is the probability density evolving over a period of time, given that this random variable is evolving according to a SDE? That's what the Fokker-Planck equation is telling you. And you can use this equation over here to get this following probability flow ODE. It was, it's a result that you can derive not quite easily, but decently well. And it was also explicitly present in this paper by Song, ETL. Yeah, and this formula over here, which has both the drift term as well as the score function, which is nothing but the derivative of the log probability. This, in essence, is the probability flow ODE equation. And this is the ODE equivalent of the SDE that induces the same marginal distribution. And so now that we've converted our SDE to an ODE, and given that we've already devised a framework for modeling ODEs better, we can just use that framework over here, given that we know what G of t is, which is the diffusion schedule. And that, in a sense, is what we do. And this is motivated by simulation-free Schrodinger bridges paper from Alexander Tong. And compared to that paper, the difference here is that instead of just using linear Brownian paths, we use spline bridges. And yeah, and in the proposition 4.4 in our paper, we also show that given that you use like a spline bridge, what the corresponding loss function that you essentially use to train the score as well as the velocity field looks like, so to speak. And so the implication of this proposition is ultimately that we can, yes, utilize B-spline construction via probability flow ODE to recover the true drift of the SDE given a constant sort of diffusion schedule G. Yeah. So now that we've seen how we are going to use B-splines to construct these conditional probability paths for modeling both ODs and SDs, what do we test it on? We test it on a bunch of things, but starting with, we will test it on a set of dynamical systems that have both a OD and as well as a SD equivalent, so to speak. Exponential decay, harmonic, damped harmonic, Lotka-Volterra, Lorentz. Okay. And these are the experimental results where we benchmark spline flow with neural OD, latent OD. MMFM is basically multi-marginal flow matching, which uses cubic polynomials to approximate the conditional probability path, whereas TFM is trajectory flow matching, which uses linear probability paths. So what we observe from this table is that by and large, spline flow is orders of magnitude better compared to the flow matching baselines. And if not, and compared to latent OD, which is usually like the state of the art for modeling irregular dynamics, it's at least on par with it, while being significantly or dramatically faster, as we'll see in the corresponding slide, future slide. P here is basically the observation probability. So P of 0 means that you observe everything. P of 0.75 means that you've removed 75% of your data. And this slide is basically the speed up of spline flow with respect to latent ODE. And as you can see that it's orders of magnitude faster than latent ODE while being comparable in performance, if not better. And this is also the visualization of Lorentz chaotic dynamics. So Lorentz system is a chaotic dynamical system that's usually used for weather prediction models. And the chaoticness here results from the fact that it's extremely sensitive to the initial information that you provide. So based on the initial point that you start off with, you can end up either in one attractor basin or the other, or can alternate between the two. And this is what makes these systems like really interesting to study, as well as use these systems to model weather, because even in weather you have these chaotic dynamics or butterfly effects that you usually want to sort of characterize. And similarly, we have results for the SD case across like metrics such as MSE just on the SD trajectory, Wasserstein and MMD. And even here, we observe that spline flow performs better than the Schrodinger, simulation-free Schrodinger bridge, the baseline of simulation-free Schrodinger bridge from Tong. Interestingly, we also notice that when we model higher order systems like Lotka-Volterra or Lorentz, higher order polynomials seem to perform better in our case compared to sort of like simple systems such as exponential decay or damped harmonic, where order 1 is the best performing one usually. And similarly, we also have performed experiments for cellular dynamics, and I will explain what these cellular dynamics means, which is because it's also the topic of the second part of this talk. And we've used similar data sets as in the coming talk on brain regeneration and embryonic evolution. And even here on the fate embedding, which is on, so these results are on fate embedding of the cellular dynamics, which is an embedding that's not PCA, obviously, and it's also not the full dimensionality of your gene expression. It's the embedding that is characterizing the topological nature of your data, so to speak. So it characterizes how it characterizes the relationship, what is the topological relationship of a group of cells with respect to another group of cells, and hence you also get like a curved nature of data for fate embedding. And for this case as well, we see that higher order spline polynomials are the better performing one for both interpolation and extrapolation task. Yeah, and this graph over here is basically showing you the best spline flow degree that you have versus the sparsity, so to speak. And you observe that, especially for nonlinear dynamical systems, you do see that as you have sparser and irregularly sparser and sparser data, the higher order of spline flow or the higher order with which you devise the underlying spline paths does help. Yeah. Okay, so any questions? If there are no questions, then we can move on to the second part of the talk, which is context flow. So this is a work that was in collaboration with Francesco Ceccarelli, Sean B. Holden, and Pietro from University of Cambridge, Xiao Zhang, my advisor, and Jovan Tanevsky from the Heidelberg University Hospital in Heidelberg. So before going into the details, I also want to talk about the task of trajectory inference and why spatialomics. So why do we care about spatialomics as a modality, and why do we care about trajectory inference as a task? So when you say that you want to know what's happening in a biological system, let's say you cut out a tissue sample and you want to know like what is really happening in this tissue. What does that entail? It entails basically you probing that tissue to characterize what genes are expressed in this tissue, what are the proteins present, what are the metabolites present, and what are the other small molecules present, so to speak. All of these probing mechanisms are one-time probing mechanisms. By that, what I mean is for a given set of cell or tissues, once you do probing for any of these modalities, the tissue or the set of cells die, because you really need to do some PCR, or you really need to open up the cell membrane to basically see what genes are expressed, and so on and so forth. And so once you probe a given sort of tissue or organism, that set of cells, those set of cells die. And single cell RNA-seq was like a good leap forward, in the sense that earlier you used to take this tissue and only get the bulk readout. So you're basically averaging out the gene expressions or the genes expressed across this bulk. With single cell RNA-seq, you could then say that, oh, for this tissue, I disintegrated this tissue into, let's say, 1,000 cells, and this is for individual cell. These are the set of gene expressions that I've read. But with this, the problem now is that you lose. So even though you do not get like a bulk average readout, you still do not know where this readout is coming from, because you've lost the spatial context, because you've disintegrated the tissue. Spatialomics, using spatialomic technologies, what you can do is you can read spatially resolved gene expressions, and so that allows you to retain the spatial context of the gene expression values that you're going to read, so to speak. So this is why spatialomics is a good omic modality to work with. Now, why trajectory inference? Why do we care about what is going to happen to a state of a set of cells that we are studying? The reason why this is important is because think about a tumor sample that you have. It can be sort of in a patient, in an animal, in a sort of lab culture, whatever it is. Wouldn't it be nice for you to have a system that's going to tell you, given this set of cells at this point in time that I've read out, how is this thing going to evolve in time? Because once you have a notion of how this thing is going to evolve in time, you can then devise interventions, medications, to basically let that tumor either die or maneuver the state in the future, once you extract the causal and the mechanistic know-hows of how this evolution is basically happening. This is essentially the North Star of trajectory inference problem, in the sense that you want to characterize how a set of biological cells are going to evolve over a period of time, so that we devise interventions and treatments beforehand, so to speak. Although it's a tall order, and at this point in time we are nowhere near that, this is an attempt towards that, so to speak. So our objective is basically to infer the temporal dynamics that explains the population level observations best. The difficulty, however, as with any omic profiling technology, is the fact that it's destructive. So you can't really track the cellular states over time, because the moment you read like what's happening, the cell dies. Mathematically speaking, you can characterize it as if you have a time evolving probability distribution and you're observing or taking samples from a distribution at a given point in time. And so mathematically, the task is basically to characterize these dynamically evolving or time varying probability distributions. And that's essentially what all generative modeling is all about, as we just saw. All generative modeling is about telling you, given this initial probability distribution, can I learn the set of transformations that are responsible to take this initial distribution to the final distribution that we want. That's what all generative modeling is all about. That's what flow matching does in an ODE fashion as opposed to SD fashion in diffusion. And long story short, as we just saw, the training objective finally is then that of a regression loss, where you're training your velocity field on this conditional path, conditional velocity field, so to speak. Now, for a bunch of disentangled cells, what you want to do is you want to construct conditional paths between these cells. So how do you draw that bridge? You draw that bridge firstly by just drawing straight lines, as we saw. But how do you decide which cells should I pick from this state and which cells should I pick from this stage? So often people use OT couplings, because OT couplings also have good interpretation in the sense that they represent the transformation that's kind of minimizing the energy or the work done to take a set of cells and transform it into another set of cells. And that's why people usually sort of run optimal transport algorithms on top of these two sets, get the optimal transport couplings, draw a straight line, train the flow matching algorithm on top of that. So firstly, why? So even in optimal transport couplings now, there are different OT formulations. Like there's no one good set of optimal transport formulation that you should use. So why people use entropy regularization in the optimal transport problem? The primary reason is because regularizing with this entropy enables you to use something known as sinkhorn iterative algorithm. And this is one of the algorithms that you use to train or to get or converge to the optimal coupling, so to speak. And people have devised GPU optimizations for the sinkhorn algorithm. And that's why it's always preferable that you train or get your OT couplings using this sinkhorn algorithm. And all of this is enabled by the fact of this entropy regularization term. And the reason why this is the case is for that you need to sort of write down the Lagrangian of this linear optimization problem, and once you devise the solution for the optimal coupling, you can then see why this is amenable to like sinkhorn algorithm. But I will not go into the details of that. Just know that in this optimal entropy regularized OT formulation, you can then show that depending on this epsilon, which is the hyperparameter for tuning the entropy influence, you can get either side of the sort of scenarios. When it goes to infinity, you basically get independent couplings. When it goes to 0, you recover the original Kantorovich formulation. But why is this OT formulation, or what people usually, when people usually use these OT formulations in flow matching, why is this suboptimal for the case of spatial omics? This is suboptimal because, as seen in the figure over here, what you have is two set of slices or spatial omic slices from two consecutive time steps. And here, what I've done is I took these spatial omics data, I ran sinkhorn algorithm on it, I get optimal transport couplings. And these are couplings between cells that serve completely different functionality. So for example, here, immature MSN to immature CMPN cell, this transition that's happening over here. So these two cells serve completely different functionality, and in no point in time will one cell transition into another, so to speak. And even though this is like biologically implausible, the algorithm devises these couplings to be statistically plausible and optimal, so to speak. And the reason is because the algorithm does not know that this is a biologically wrong coupling. It lacks this groundedness in reality. Okay, and that is important because eventually you are training your flow matching algorithm on these implausible couplings. Okay, and this is why it is important that you constrain your generation using domain knowledge. And that is the essence of context flow. So mathematically speaking, when you're devising these conditional probability paths, there can be multiple probability paths that give you the same observation. But you do not want to take any probability path. You want to take the path that is the most biologically plausible because you want to generalize during inference. And that's all that context flow does, which is incorporates biological prior knowledge to constrain the generation by penalizing implausible conditional probability paths. Now, the important question is, how do you incorporate this biological prior knowledge, and what is the correct level at which to incorporate this biological prior knowledge? So before even going into how we incorporate biological prior knowledge, we should also think about what is the correct way, what is the right biological prior knowledge in the case of spatial omics for us to incorporate. So we go with two kinds of prior knowledges. One we call spatial smoothness, the second we call cell-cell communication pattern. So both of these priors are derived from the fact that effects in biology move smoothly and not abruptly, so to speak. So a group of neighboring cells are going to have similar expressions within themselves compared to a group of cells that's like physically far away. And that's in essence what this spatial smoothness prior is trying to capture. The cell-cell communication pattern is trying to capture the fact that cells do not live in isolation. Every single cell is communicating with its neighbor, and those neighbors are communicating with their neighbors. And this communication is happening via chemical pathway signaling mechanisms, which at any given point in time, a cell is basically communicating the fact that it needs XYZ metabolite, protein, or whatever. And that's why it sends the corresponding ligand further away in the neighborhood to sort of get what it needs at that point in time. And these communication patterns you can characterize using ligand receptor interactions. And it's plausible that if two set of cells in consecutive slices have similar communication patterns, then their transition is kind of more plausible than otherwise. And similarly, if two cells have average neighborhood expression that's similar to each other, their transition should be more plausible than otherwise. In our case, the spatial smoothness is kind of acting as a proxy for the actual spatial coordinates. And the reason why we do not use actual spatial coordinates is because we do not have a global frame of reference, so to speak. So that's why we devise this proxy in terms of mean neighborhood expression. And then once you know that this is what plausible coupling should look like in the case of spatial omics, we encode that knowledge into transition plausibility matrix, which is basically telling you, given my cell K at time I and given my cell L at time I plus 1, what is the plausibility of this transition happening without running any kind of optimal transport or any kind of learning algorithm? Like, biologically speaking, what does my intuition tell me? How likely is it for this transition to happen based on spatial smoothness prior and cell-cell communication patterns? That is what's encoded in transition plausibility matrix. And then what we do is we encode this plausibility matrix into the optimal transport formulation. And we propose two novel prior aware OT formulations to encourage biologically plausible velocity fields. So on the left, we have included this transition matrix in the cost term. On the right, we have included this transition matrix in the entropy term, so to speak. The left term is a harder constraint in the sense that it's telling you the fact that the transition between cells K and L should not happen if they have a higher sort of cost in terms of how similar their how different their gene expressions are, but also how implausible do you think this transition is based on your prior knowledge. So you incorporate all of this directly into the optimal transport cost function. In the entropic term, in the entropic formulation, what you do is you use this plausibility matrix as a soft regularizer, so to speak. And in practice, we observe that this soft regularizer performs better. Not only does it perform better, it also has, it also retains the computational advantage of flow matching, because you basically get a transformed Gibbs kernel. And this Gibbs kernel is the entity that's used in synchron iterations, so to speak. So all you do in this second formulation is you get a new Gibbs kernel that is biased based on the transition plausibility matrix over here. And you can just then use your previous synchron iteration pipeline to train your, to get the OT couplings and to sort of devise your synchron iterations. And this is important because this tells you that you are kind of nudging your couplings ever so slightly towards your prior knowledge, but you are also not just relying on that, but rather it's a dot product of your knowledge as well as your cost, which is the e raised to minus c by epsilon term over here. And so this is kind of a softer bias compared to the fact that you encode everything in your cost, so to speak. Yeah, and so given that we do both of these entropic as well as cost modifications to the optimal transport formulation, what do we train it on? As we saw in the case of spline flow as well, we evaluate these algorithms on 2 data sets, or we actually evaluate on 3 and more data sets in the paper, but here I will just present 2. So first is the axolotl brain regeneration data set, which is basically a spatially resolved data set for salamander brain that was injured and how that injured brain is sort of healing over a period of time. And the second is mouse embryo organogenesis, which is basically telling you how is it that a mouse embryo evolves over a period of time into different organs, so to speak. So these are our data sets. For evaluation and for sampling, we use 2 sampling criteria. So first is next step sampling, and the second is initial value problem sampling. So initial value problem sampling is basically telling you, given that you want to know how this set of cells is going to look at day, let's say 5, I will start from day 0, and then I will integrate the velocity field over the entire time horizon and then give you the final result. This is a much harder test of generalizability because you need to ensure that the intermediary velocity field that you've learned across the time horizon is correct, and not just the time period before your sort of required time point, which is what is happening in next step sampling, which is just taking the previous observation and just integrating that forward. And before going into the flow matching performance, I just wanted to comment on the OT performance. So here, as we see, the context flow OT formulation that we have results in significantly few biologically implausible OT couplings for this inhibitory and excitatory linear switches, as we saw earlier. And across like different metric values and across different sampling methods like next step, IVP, and for different, and across like interpolation and extrapolation, where we evaluate on the middle held out time step and the last held out time step, context flow performs better than the baselines naturally. And not only do we perform better on metrics, we also retain the biological composition over a period of time. What do I mean by that? So given that you start with the initial set of cells, those cells are going to transform into other cells as they evolve over a period of time. The point that we want to make is that over a period of time, we also preserve the biological composition of these cell types correctly. Because from the data you can know that the later half of the time process will have more mature cells like mature liver, mature kidney, mature heart cells compared to the immature cell types. And we want to check, do we observe these like biological sensibilities in the prediction that we make? And that's why for doing that, we calculate basically the histogram of cell types for the ground truth and generated case, and we check the KL divergence between the two. And in the case of context flow, we perform better than the baseline. And then that in a sense also concludes our evaluation part. And so in conclusion, I just want to say that if you understood nothing by the end of this talk, at least for context flow, understand this. In the cases where you do not have enough data to infer the true underlying scientific principles from the data, it's very important that you ground your generation in the knowledge that you already know about the system. And context flow and spline flow are one way of doing that. And although these are not the only ways, here I have basically given an exposition on the fact that all of your knowledge should be encoded in you devising good conditional probability paths. There may be other ways, but this is one of the way. And so like if you do not have tons and tons of data for you to truly characterize what the true underlying principles of the system are, it's always better to sort of ground them in what you know, so that the learning algorithm doesn't do the hard part of redoing and relearning those principles again correctly. So in conclusion, context flow offers a generative modeling framework to infer biological plausible temporal tissue dynamics by incorporating prior domain knowledge. And more specifically, we do so by utilizing tissue microenvironment and communication patterns to constrain bias degeneration towards biological feasibility. And with that, thank you. Questions? So the question is, can we use branched processes or branched diffusion processes to model these systems? And I think you can use them, but they're not going to give you like a dynamical nature of things. So in branched systems or branched diffusion and things of that kind, you often start with the initial state and then you're going to a set of states, so to speak. So you start, for example, in the case of protein conformation, you start with the initial protein configuration and then you devise where all can you go from here. But it's not giving you a dynamical view of things, like how should the velocity field and the corresponding score or whatever it is, that should look as a function of time, so to speak. It's just telling you probabilistically, given that I'm here and given that I'm here based on some Boltzmann distribution, this is what my future states should look like with whatever probabilities. So yes, I think